Hi everyone, thank you for joining today. This presentation will cover a brief introduction to single molecule localization with the Vutara system, and then focus on multiplexed imaging strategies, including DNA paint and oligostorm. Biological systems exist across a wide range of sizes. Light microscopy, particularly fluorescence microscopy, has been a useful tool for visualizing biological organization. However, traditional approaches are unable to resolve features below about 200 nanometers due to Abbe's diffraction limit, meaning we're unable to distinguish objects that are less than 200 nanometers apart. This is rather unfortunate because cells, including genomic structure, are highly organized at the nanoscale, and structure often informs function. To be able to visualize structures well below the diffraction limit using light microscopy, a variety of super resolution techniques have been developed. One such method, which will be the focus of today's talk, is single molecule localization microscopy. In recent years, single molecule localization microscopy has gained a lot of traction in the field of genomics, which focuses on understanding the complex organization of DNA. Within every human cell, roughly 2 meters of DNA is packed in a 5-micron space, the nucleus, in the form of 23 pairs of chromosomes. The estimated 20 to 25,000 genes that make up these chromosomes are responsible for the production of all the proteins in the human body. So understanding the structure, function, evolution, and mapping the genome is essential to discovering the genetic basis for health and disease. Knowing the genetic composition of the genome is important, but establishing how genes are organized and interact with one another in their environment is also important. Being able to visualize organization and structure on the subchromosome level is necessary for understanding these relationships and how genes function. Visualizing genomic structures at this level can prove difficult because of such a large number of targets within the genome. There are not enough chromatically separable probes available. Luckily, new approaches allow for sequential labeling of targets using the same fluorescent probe for imaging. In October of 2020, we released our latest platform in the Vutara line of super resolution microscopes, the VXL, offering a much smaller footprint than previous models and optimized imaging capabilities. The Vutara platform for single molecule localization with integrated fluidics makes these sequential imaging applications feasible for a large number of targets that spatial genomics studies require. The Vutara offers a proprietary biplane technology and is the only commercial single molecule resolution system offering this technology and the capability of imaging far from the cover slip. This is due to the fact that biplane detection does not rely on point spread function engineering to obtain Z information. Other systems that rely on turf and high-low illumination are limited to about 5 microns of imaging depth with a difficult three-dimensional calibration. And this gives the Vutara a huge advantage, allowing imaging of the entire thickness of a cell, cell colonies, and tissue sections and model organisms. The flexibility the Vutara gives you in terms of sample type makes it suitable for a variety of biological functions. For the remainder of the talk, I will focus on multiplex imaging related to DNA paint and genomics imaging using oligostorm and ORCA. In single molecule localization, often referred to as SML, if we take a diffraction limited object and label it densely with fluorescent molecules, what we see by conventional light microscopy looks like a diffraction limited blur regardless of the underlying structure. With SML, these molecules behave in such a way that they're able to turn off and then back on one by one, so that the center of each position of each molecule can be recorded during an image acquisition. At the end of an acquisition, all these points are used to reconstruct a final super resolution image. In practice, data collection looks like this, where you see a field of view with blinking molecules, ideally no more than one per diffraction limited area. As a reminder of localization microscopy methodology, the fluorescent on state is optically controlled, so only a small subset of the molecules is on at a given time, and this results in point spread functions being sparse across a single camera frame, so they can be isolated individually. After identifying individual point spread functions, localization algorithms are performed on each point spread function. This allows us to extract the below diffraction limit spatial information on the location of each molecule. The super resolution image is then a result of compiling all the localized information from each point spread function. Here we see a single point spread function being identified at which point it is fit with the localization algorithm, and its center is identified and used for the final super resolution image. The optical design of the Vutara takes localization one step further through the use of biplane imaging. Here we have the standard laser lines with an optional 750 nanometer laser coupled into a fiber which goes into the excitation path of the microscope. While imaging, we have a V-focused drift correction system containing a separate 850 nanometer laser that bounces off the cover glass and is recorded on a separate camera. Any shifts in Z are recorded as a lateral shift of this beam and automatically corrected by a piezo during imaging. The emission path contains a spatial filter to remove any out of focus light. After passing through the spatial filter, light enters a 50-50 beam splitter that sends half the light to focal plane 1 and the other half to focal plane 2. So every acquisition with the Vutara contains three-dimensional information because of its biplane detection path. If we look at a molecule in X and Z, we see an elongated PSF that looks like this. If we then visualize the X and Y plane at two positions, you get two different point spread functions which can be used to generate a table that contains Z positional information. This table can then be used to identify the Z positions of other molecules. For example, if a molecule is located at the focal plane, both planes will be in focus. In contrast, if the molecule lies above the focal plane, its two resulting point spread functions will look like this, and we can identify the Z position as a positive 250 nanometers. And now when we do a reconstruction using the Vutara, we simultaneously image two focal planes so that we can localize both the X, Y, and Z positions. The advantage of this is that unlike other commercial systems that use the point spread function engineering to get their Z information, it's much less sensitive to imaging deeper into samples. So how do we get an isolated source in a densely labeled sample? There are two main methods for obtaining optical control over fluorophores. You can use fluorescent proteins that switch from off to on with UV light, or ones that shift emission spectrum. The second, more widely used method is the use of organic dyes, which through a series of light-driven reduction reactions bind a side group to the fluorophore, rendering it non-fluorescent. An alternative to these methods that rely on photo switching of organic dyes to generate blinking is paint, which stands for points accumulation in nanoscale topography. Here, blinking is generated from the transient binding of a fluorophore to its target. Standard D-storm labeling uses primary and secondary antibodies labeled with a photo-switchable fluorophore. The antibodies remain fixed on the sample and photo-switch given the correct photochemical environment. With DNA paint, the generation of single molecule blanks is a bit different. Blanking is generated by the transient binding of a fluorophore to the target. When it binds, we see it on the camera and we can localize the blank. It gives you a lot more photons, so the localization precision is much higher, but it's usually much slower imaging, scales of 5 to 10 times slower than standard D-storm experiment. Although there have been recent advances in this technology to reduce that time dramatically. Here we see antibodies labeled with a DNA barcode in yellow and a solution of complementary oligos bearing a fluorescent tag. These probes are short enough so that during the course of imaging, they bind and fall off the DNA barcode, allowing bound probes to be imaged and localized. When comparing the two methods, both have their advantages and disadvantages. D-storm imaging is fast, but there are a few dyes that photo switch really well. DNA paint is great in that regard because the fluorophore used does not have to photo switch. Therefore, you can choose a dye with a high photon budget, which results in a high localization precision. The drawback, however, to DNA paint is often the imaging time because it relies on diffusion of probes. Typical imaging times are much longer compared to D-storm imaging. You might be familiar with this paper by Ralph Youngman, where DNA paint is used in a way that allows sequential imaging of multiple targets. Here, multiple cellular targets are labeled with antibodies bearing a unique DNA barcode for each target. In the first round, complementary sequences for the first structure are applied to the sample and imaged. They're washed out and the sequence for the next structure is applied and imaged. This process repeats until all the structures have been imaged, resulting in an overlay of all 4 structures. Here's an example of a DNA paint image taken on a Vutara system. It's a single color experiment showing extremely high resolution images of microtubules. And this sample was labeled with commercially available secondary antibodies purchased from Massive Photonics, a company specializing in creation of DNA paint probes. To extend the method further, we imaged 2 color DNA paint samples on the Vutara. Here's an image of microtubules in clathrin, demonstrating that paint multiplexing is possible using the Vutara system. These samples were also labeled with oligos from Massive Photonics. And here's a scan through of that sample. Second example, or rather third example of DNA paint in a 10 micron slice of retinal tissue sections, imaged using the Vutara 352. And here's a scan through of that tissue data. A second multiplex imaging strategy is oligostorm, which is the application of oligo paint probes to image the genome. This technique combines the multiplexing capabilities of paint with the speed of D-storm imaging. Special probes made of DNA sequences that complement a genomic region can be designed to label specific genes. These probes also bear a region for binding the fluorophore-labeled DNA strands that serve as imaging probes. The regions can bind longer DNA sequences for use in oligostorm imaging and shorter DNA sequences for oligodNA paint. The difference in the two methods being that oligostorm relies on blinking of the fluorescent molecule to generate localizations, where DNA paint localizations would come from the transient binding of DNA onto the sample. Having both options available allows for the same region to be imaged twice if necessary. After developing a set of oligo paint probes, they can be imaged sequentially. The sample starts with all the main probes containing the genomic homology bound to the sample. In the first fluidic cycle, a set of complementary probes is flowed in that bind to the first barcode region. The region is then imaged using single molecule localization. Afterwards, a new set of probes is flowed in, which remove the previously bound set and introduce a new set that will be imaged. And then this cycle repeats until all the barcodes in the sample have been imaged. This oligostorm technique allows direct visualization of chromatin structure in situ and has been used successfully to perform multi-chromosome walks in this paper using the Vutara imaging platform. Direct visualization and correlation with sequencing of chromatin provides a more complete understanding of nuclear functioning. The broader workflow of these experiments looks like this. A library of many thousands oligos is designed and oligostorm probes are generated and labeled onto a sample. The Bruker Fluidics platform is then used to label the probes individually so that each probe can be imaged by single molecule localization with the Vutara. From there, data is drift corrected and analyzed using SRX software so that the final image can be assembled for each genomic walk. This workflow has achieved optical resolutions of around 25 nanometers in X and Y and 110 nanometers in Z. At first glance, these experiments can appear very noisy. So how do we go from this very messy image to isolating signal from genomic structure? We use the data associated with every localization to separate signal from noise. We have a few algorithm options in SRX for performing cluster analysis, but the one used for genomic data I've showed you is dbScan, which stands for Density-Based Spatial Clustering of Applications with Noise. And this algorithm, there are two key parameters, epsilon, which specifies the radius of a neighborhood with respect to some point, and min points, which is the minimum point that are required to form a cluster. In this diagram, min points equals 4. Point A and the other red points are considered core points because the area surrounding these points in an E radius contains at least 4 other points, including the point itself. Because they are all reachable from one another, they form a single cluster. Points B and C are not considered core points, but are reachable from A by other core points, and thus belong to the cluster as well. Here, point N is considered noise because it's neither a core point nor is it directly reachable from other core points. Once clusters are identified, metrics are then able to be calculated. In this view of our SRX software, you can see a table of the different clusters and metrics calculated, such as the number of particles in a cluster, volume and surface area, sphericity ratios, density of particles, and radius of gyration. Another feature of this software allows you to highlight a single cluster in a list and have it appear highlighted in the visualization window, letting the user seamlessly associate the visual spatial profile with quantitative metrics. This video shows a 13-step walkthrough of chromosome 19. Because of the sequential nature of the experiment, the same fluorescent probe, Alexa Fluor 647, which happens to be the best fluorescent probe for single molecule localization microscopy, was able to be used for each round of imaging. Analysis in SRX enables you to generate in situ high C maps, which exhibit strong correspondence to ensemble high C maps, indicating the validity of image-based measurements and overall capability of oligostorm to elucidate the organization of chromosomes. Furthermore, oligostorm data and analysis in SRX can be combined with ensemble high-C data to generate 3D genomic models at resolutions of 10 kilobases, despite being imaged at much larger step sizes in a technique known as integrative modeling of genomic regions, otherwise known as IMGR. Altogether, our system enables you to get images like this, which allows you to perform experiments ranging from collecting data on multiple chromosomes down to visualizing small subfeatures of loops. So not all researchers looking at the genome are necessarily interested in doing large walks like in the previous example. In this example here, studying centromeres during interphase, a smaller region, 1 megabase compared to the 8 megabase walk on previous slides of a centromere was labeled and imaged with the Vutara. From the densely labeled data, you're able to draw a trace of the linear sequence to visualize it spatially. The workflow of oligostorm also works nicely for fluorescence in situ hybridization experiments. Here is a two-color fish example looking at interaction between two different topologically associated domains. SRX, the software that comes with the Vutara for acquisition and analysis, allowed researchers to visualize overlap of the domains and calculate metrics about them. In a similar manner, the system can be used to perform RNA fish experiments. Here's an example data set from Brown University showing early passage normal diploid lung fibroblasts labeled against non-coding RNA MALAT1 using a set of 48 Stellaris fish probes. SRX software is fully equipped to handle a large range of analysis, from looking at spatial distributions to co-localization studies. Here's a view of the user interface displaying a cluster analysis histogram. Once the localizations are assigned to clusters, then metrics can be calculated. Here on the right is an isosurface rendering of a topologically associated domain in blue and a compartment in magenta. From this data, it would be possible to further evaluate the clusters by calculating metrics such as overlap volumes or fractions. Our most recent project has been working to integrate a workflow into SRX known as Optical Reconstruction of Chromatin Architecture, also known as ORCA. ORCA uses oligopaint like oligostorm does, but rather than image at the single molecule level, it takes subdiffraction images at the wide field. All the fluorophores in a single genomic region are imaged as an ensemble rather than stochastically. The center of mass is determined from the image, and an ORCA walk is generated by connecting the center of masses. The technique utilizes small probe step sizes and is ideal for looking at smaller regions or single genes. Here's an overview of the ORCA workflow. A set of oligonucleotide probes is designed to label a genomic region of interest, much like that in OligoStorm. Each segment carries a unique barcode and represents a single step of the ORCA walkthrough. Segments are imaged sequentially to build a 3D reconstruction of the chromatin architecture. The 3D position of each segment is then used to build a distance map that's comparable to a Hi-C map that is typically used in genomic studies. In practice, this image shows a wide field image in SRX where the boxes indicate identified probes. A representative 3D reconstruction of chromatin architecture image is then generated by SRX software by connecting all the identified segments during each round of imaging. For each cell, a distance map is created from measuring the distances between each segment, and then a contact frequency map is generated in SRX software by averaging the distance maps associated with ORCA walkthroughs of all the cells. And then this data can also be exported in standard formatting associated with Hi-C data so that the data can be viewed in other external softwares other than our own. If you compare STORM, OligoStorm, and ORCA, decreasing the step size increases the sequence resolution. With STORM, you get an overview of the shape of the structure without any sequence information. OligoStorm adds in the sequence context to the structure. And with ORCA, you get a finer sequence resolution, but lose out on the space filling component that you get with OligoStorm. The techniques are complementary, and their use really depends on experimental capabilities, as well as the target features being studied. If your desire is to do a high throughput study of a small genomic region, then ORCA is a great choice. Whereas if your desire is to take larger steps and image a much larger genomic region, then OligoStorm would be preferred. No matter which serial labeling strategy you choose, automated fluidics is essential due to the large number of targets to be imaged. We offer a fully integrated fluidics unit designed with single molecule imaging in mind. Each unit has 4 large reservoirs for high volume reagents like imaging buffers and washes, and then 15 smaller reservoirs for those precious labeling reagents. These smaller reservoirs may also be swapped out during imaging, making imaging greater than 15 targets possible. The user interface is intuitive and gives the experimenter full control over fluidic sequences and parameters. The unit can be used as a standalone with an existing microscope or fully integrated into the acquisition process using a Vutara microscope and SRX software. The Vutara VXL and SRX software provides a powerful system for 3-dimensional single localization microscopy of a wide range of sample types. The ability to integrate fluidics with the system makes multiplex imaging applications much more accessible, and SRX software that comes with the platform provides an all-in-one solution for data acquisition, visualization, and analysis. That concludes my presentation today. If you think of any questions or comments in the days that follow, please feel free to email me. My email is here. And also feel free to visit the website for more information on the Vutara if you're interested.